Hello, my name is Anuj Pawar, I'm a zoology major at SIU, and today I'll be talking about the applications of life science in space. So, what is life science? Life science encompasses everything that is organic, which is you, me, your pet dog, cat, your fish, plants outside that produce air, what else? Cows, which produce milk, uh, and everything that's natural in the world, pretty much. And so when integrating life science or biology with space, it's actually very much needed. The reason for this is because uh, we have to see what the effects of space traveling are on humans and how we can better uh, produce space travel for our astronauts and for future explorers as well. So, as stated here by NASA's space biology overview, is that their purpose is to build a better understanding of how spaceflight affects living systems in spacecraft, such as the ISS, International Space Station, or in ground-based experiments, and to prepare for future human exploration missions far from Earth. Now, when doing so, they are going to have to focus on what is the issue and then what is their solution. So in this presentation, we'll be discussing animal test subjects, microorganism studies, plant food production, and applying knowledge for future space explorations. Now, to know more about space biology, we're going to have to go back to the beginning of uh, space exploration. So the beginning of space exploration was around nearing the end of World War II or at the end of World War II with the space race between the United States and Russia. And so, which was around the 1940s, mid 1940s. So initial spaceflight tests used animal test subjects to determine whether or not or to determine what were the effects of high velocities or g-forces were on a organism body, what the effects of altitudes were on the organism bodies, what the effects of space were, because it's an extraterrestrial environment. It's an environment that our bodies are not suited for. So in order to see what would happen, they would send animal test subjects, large organisms. So one of the first significant uh, of these animal tests were in 1948. So around late 1940s, there are these V-2 rocket tests, which were directed by one Captain David G. Simons, who was at the Aeromedical Laboratory at White Sands. Under his watch, well, there were multiple tests. First 5 biological flights had failed for multiple reasons, uh, but due to these failures, they had to produce a capsule in order for there to be additional protection. Going back to the 5 biological uh, failures were that they failed in live recovery of the animals. Their tests for rockets were probably successful. So with the capsule and extra protection, June 18th, 1948, their test subject named Albert, a rhesus monkey, was placed into the capsule and ready for launch. There is no actual, I could not find an image of Albert, but he was a rhesus monkey, so on the right is a contemporary photo of a rhesus monkey. And so for Albert, uh, sadly, Albert died before the launch of the spacecraft because there were respiratory issues due to being in the capsule, which was very narrow. But if he had survived, he would have died anyways because the parachute failed upon re-entry. So there were multiple operation failures, but this caused scientists to focus in on what the issues were and what they could do to better future space flights or future tests and better future rockets, which leads to human travelers in space now. Having safe flights and having healthy lives. So these future tests then start to look more into internal physiological effects of spaceflight, heartbeats, respiration, neurological effects of spaceflight, etc., etc., etc. And so the initial tests uh, basically revealed like information that better the future because now we're having better space flights, better uh, yeah, basically better space flights. You know, people aren't dying left and right, or astronauts aren't dying left and right when they're going up in space. And it's all thanks to the animal test subjects that were used, the mice, the rats, the monkeys, the dogs, the cats. I don't think they use cats, but no, the dogs, like Laika, in Sputnik 2. Uh, so thank those animals for giving their lives to science. So from large living organisms, let's go to more small scale organisms. Microorganisms. Microorganisms are everywhere. They're in the forms of bacteria, viruses, uh, little itty titty, little tiny multicellular organisms like mites on your lashes. They are everywhere. As though, as creepy as that can be, it's alright. Most or some of them are pretty much harmless. There are multiple uh, microorganisms, bacteriums in your gut, which actually help in breaking down certain foods and producing nutrients and stuff that we actually absorb. So I mean that's a good thing. So there's also like what is it? It's a gut microbiome that's in your gut. Microbiomes in your gut that helps. So point being, microorganisms are everywhere, which also includes space. So at the ISS, International Space Station, it's a closed environment. A closed environment is something that is secluded from an outer source. So since the ISS has multiple astronauts within the station, uh, so people can leave a bit of a mess here and there. Of course, there's dust. There's a whole bunch of stuff up there, so they have to be very careful. And so, yeah, it's like an enclosed environment. A good example would be a microgravity space glove box, which I believe I actually talked about in a previous presentation. So that would be a good example of a closed environment because it's separated from an outer environment. And so, because people can leave a bit of a mess when they're disposing waste, they actually have to send down air filters and vacuum cleaner bags to the uh, back to NASA, uh, so that the scientists can take a look at what little things are there. And so, the reason for this is so that if there were to be any new organisms, they can determine whether or not if they are harmless or if they're harmful. If they're harmful, they can extract a vaccine or some sort of cure. And so, interesting that happened recently, I believe within the last couple of years, was Dr. Kasturi Venkateswaran at NASA's Jet Propulsion Lab discovered a new species of bacterium, uh, dubbed Solibacterium Bacillus calami. And what he found very interesting about this organism was that it possesses an extraneous layer, an extra layer rich in lipoglycoprotein, and this type of protein is actually used as a protective layer against radiation. And very interesting. So what he extrapolated was that if they could, I think a good term would be mass produce, because these proteins can be small. If they can mass produce this protein, it could be used as an agent for as like an extra protection layer in spacesuits for astronauts and give them uh, some extra protection against space radiation when they're out on moonwalks, when they're farther away from Earth because Earth has a because Earth has a magnetic field. There's no need for that type of protection because the magnetic field. No, the ISS is within Earth's magnetic field, so there's no need for that. But if let's say they would go to the moon or go to Mars, they would actually need that type of protection. Why? Because well, they're placed out there, and they can get radiation. So that extra protection layer can be very helpful to them. Another thing to talk about is tardigrades. Tardigrades, or water bears, are pretty much well known. They are, and what is known about them from the general public is that these little guys are tough. They can survive pretty much anything. And so, as a zoologist, this is something that I've actually learned in a class recently, animal diversity, with over this last semester with Dr. Frank Anderson at the zoology department. I'm giving him credit for all of this. And so this is pretty much what he had on his lecture slide. That when the environment is actually unfavorable, not favorable, that's a typo right there, unfavorable, they form a ton, which is a resting stage, and they dry out. And so when they dry out, like water just leaves their body. It is a form of cryptobiosis. I apologize. All of this is a form of cryptobiosis. And so continuing on with the ton stage, what causes so the tardigrades have the ability to just drain out their body of water, and with water out of their body, how do they pretty much survive? They have a sugar known as trehalose, which maintains their cell membrane, so their cell membranes are fine. But in this ton state, they are also in like kind of like say kind of like a hibernation or sleep mode. But in this ton state, they can survive immersion to liquid nitrogen, boiling water, high levels of O2, acids, alcohol or X-rays. But then they can also survive exposure to outer space, which is a very incredible thing. And then when their environment is favorable again, then they can absorb water and they leave the ton state, and then they just float around, do their little itty bitty things. And so when we're looking into more, entering this knowledge with this microorganism, with the lipoglycoprotein, and integrating what tardigrades are able to do, we can one add in a protective layer to spacesuits for astronauts when they're exposed to radiation, and two, for future long endeavors of travels, let's say from our planet Earth to probably the moons of Jupiter or Neptune or Uranus or Pluto. Those would take years, and so what might be easier for them to do is go into a ton state, see how we can utilize sugar trehalose to maintain the cell membranes, and what would be the processes of bringing back people from this type of hibernating weird state. Continuing on, space plants. So, what we do with plants is we eat them. We eat them because they're food. Food is anything that is a nutritious substance people or animals eat or drink, or that plants themselves can absorb in order to maintain life and growth. Plants absorb, uh, they absorb energy from the sun, they absorb minerals from the soil, they absorb CO2, and all that goes into making glucose, which is what they use for energy. For us, we as animals, as omnivores. Well, let's specifically go with herbivores because this is about plants. For herbivores, they take in plant stuff and feed upon their nutrients or their like glucose or anything, fibers, anything else that they have in there. And so, the question would be, why plants? Why focus on just plants in space? Why not have meat? Why not have meat in space? Well, the reason for that is that it's a lot more sustainable to have plants and uh, plants in space. But that's a different topic for a different slide. Continuing on to like food stuff. Uh, the type of foods that they have in the International Space Station, they have your typical types of foods. They have brownies, macaroni and cheese. There's also condiments, ketchup, and salt and pepper. Uh, but for salt and pepper, uh, those have to be in a liquid form. The reason for this is because, well, the thing about salt and pepper is that they're just one little grain of salt and pepper. I mean, one little grain of those things, they're very tiny and they can easily flow around because they're exposed to microgravity, which again causes items to flow around. And so, again, going with the example of salt and pepper, if a few of these grains were to float around, they would basically it would cause a mess. It would could get into any technical equipment. It could get into an astronaut's eye or their nose, and it could cause issues, health issues to them. It could cause, let's say before, damages to very sensitive equipment and cause a lot of issues and can cause a lot of havoc. So that's one issue. Hopefully they can solve an issue to microgravity in the future, far into the future. But going back to eating plants, plants are very easy to grow and they're very sustainable. So that's why there's a lot of focus on plants rather than meat. So NASA's Vegetable Production System, also nicknamed as VEGGIE, um, their goal is to provide sustainable, easy to grow plant food sources. So in this class, we also had a few students discuss about what were the benefits of water systems and soil systems and mineral systems to provide for making plant food. So this is kind of something that NASA has been looking into as well. In their recent project, uh, they the recent project was to improve pick and eat food capabilities in microgravitational conditions. And so their current project focuses on dwarf tomatoes and leafy greens. For dwarf tomatoes, they're Tokyo Bikana Chinese. No, sorry. What? Oh yeah, yeah. Sorry. For leafy greens, Tokyo Bikana Chinese cabbage requires a faster release amount of fertilizer within over a 100-day period, and increased red light exposed to them, and they were able to grow pretty well. Now, for the dwarf tomatoes, they use red robin tomato, which requires slowly released fertilizer over a 180-day period, but alongside a slow release fertilizing were also an increased red light exposure. And so those were the two things that they found out about those plants. And with that, they hope to utilize that for the ISS and then continue forward and finding more plant-based foods that can be used in space. Now, some issues with this are that there may be some possible stressors on the plants or on the individual eating them, or there might be some behavioral health components that uh, might need to be foreseen or studied more upon. But going back to uh, space plants and current foods, that there are these issues that have to be solved, like which have been solved. Like the salt and pepper, they simply made it into a liquid form because liquid can turn to like little balls, and you've seen those videos, little balls, and be compensated like that. And they're finding more sustainable ways to have food instead of continuously sending up other types of food, like meat that's not very sustainable, uh, which is also another issue that we're having here on Earth. And so we come to the conclusion of this presentation. What would be what were the contributors to the animal test subjects, the finding of microorganisms, especially in space, and utilizing microorganisms' abilities or animal abilities? Space would be the utilization of plants in space, as well as all has to come down to human intuition, the scientific theory, and observational constraints. Whatever we can observe is what we're focused on right at the moment. So right now we're always only focusing on uh, plant food stuff, and in the near future, hopefully we can also figure out how animal abilities like tardigrades in tongue state or organism abilities like the lipoglycoprotein on the bacteria can help out, help or improve future space exploration. Other subfields have been helpful, like besides space, space is one field of study because it requires multitude of things like astronomy um, and rocket science. But then again, rocket science also has to integrate chemistry, physics. Uh, Space travel has to integrate in biology because they have to focus on how human explorers can survive in space. And then so now here comes the question: what does the future hold for space exploration? Oh, it's a very broad statement or a very broad question. There's many things that the future holds. Right now, it just has well, future of space exploration has to depend on what ideas come up from looking at other fields, because space is just one little thing in our entire life. There are in biology, physics, chemistry, uh, biochemistry, so many other things that can go into space exploration that can benefit space exploration, make space exploration easier or even better for future explorers. And right now, one good idea on this, or one good example on this, is the Lunar Gateway project, in which they will be setting up a space station, which will be orbiting our moon. And with the Lunar Gateway project, can use everything that has been learned and taught uh, up till now, such as one thing that we know is an issue is exposure to space radiation, because in orbit of the moon or on the moon, uh, there's going to be a lot of space radiation exposure. And so they're going to have to figure out how to deal with that. Either they can make some sort of energy field themselves, or as what we've discussed right now is using the lipoglycoprotein as an extra layer of protection. And then also food sources, because it costs a lot of money to send food to them. So how can they sustain with food? They're going to have to use plant-based food. And so just as a way, 80 years ago, that they studied or used animal test subjects and studied animals and used their data from those tests to better future space explorations, are we now looking at current research and focusing in on what can better the future, the future of the Lunar Gateway project, the future of the Mars missions, and wherever else future generations decide to explore. And with that being said, thank you for listening on this presentation. It has been very long. It is about 26 minutes. And thank you, Dr. Lefticariu. Thank you for teaching this class.